In this lecture, we will talk about some special senses. First, let's define the difference between special senses and somatic senses. So somatic senses are sensations that are coming from skin as a touch, joints as body position, muscle as muscle contraction, connective tissue as a stretch. Temperature and pain fall in that same category. These sensations can be identified by mechanoreceptors for touch or pressure or vibration, by thermoreceptors that will identify temperature, by proprioceptors that will identify position, and by nociceptors that will identify pain. Special senses will come from eyes, ears, nose, or tongue. So eyes, via photoreceptors, enable us to see. Ears, through the mechanoreceptors, enable us to hear and to maintain balance. And nose, through chemoreceptors, enables us to smell, and tongue, through chemoreceptors as well, enable us to taste. When sensation stimulates constantly, it leads to the process known as habituation, when you are not aware of that particular sensation. A good example would be your clothing that touches your skin all the time. And since it touches your skin all the time, you are not consciously aware of it. You experience habituation. Special senses obviously are important to protect us so we can reach reproductive age. For instance, we should be able to see and hear the danger. Also, some adaptations were a little bit more complex. For instance, colored vision allows us to see the fruits that are ripe and suitable for eating. So first, let's talk about the olfaction or smelling. Olfaction or smelling happens in the nasal passage, in the nasal cavity. The chemicals, airborne chemicals, are dissolved in the mucus. And while being dissolved in that mucus, they interact with the sensory receptors on the olfactory neurons. When a molecule of the odorant, that airborne chemical dissolved in mucus, binds to the receptor on the olfactory neuron sends a signal to the olfactory bulb and then via olfactory tract to the brain. Important feature is that one chemical can bind to many receptors, and one receptor can sense many chemicals. Therefore, each chemical will stimulate its own unique set of receptors, meaning that olfaction works very much like an accordion. Smell stimulates certain parts of the brain that belong to the limbic system. You may remember from the previous lecture that limbic system is responsible for our emotional responses. Therefore, smell triggers those emotional responses, and often your memories or like some smell, some odors can make you sad, or some odors can smell terrible and be worrisome, like a skunk, and some odors are very enjoyable. In very close connection to smell sensation is taste sensation or gustation. So receptors for the taste are located in so-called taste buds. Here you can see a structure of a typical papillae. If you will stick your tongue out in front of your mirror and look, you will find multiple papillae, fungiform papillae, filiform papillae on the sides of the tongue, foliate papillae, and valate papillae. Inside of this papillae are taste buds, and in each taste bud you're going to find gustatory cells with the taste hairs that contain receptors. When you taste something, the chemical gets dissolved in the saliva, and this dissolved chemical interacts with the receptors on the taste hairs. That interaction triggers the nervous signal that sends the information to the brain via various nerve endings. There are five modalities, 5 categories of taste: sweet, sour, salty, bitter, and umami. So sweet taste is triggered by sugars. Essentially, sweet taste allows you to taste energy-rich food, carbohydrate-rich food. Salty taste is triggered by salt, like table salt. It's necessary to identify electrolytes in the food. Sour taste is triggered by acids. It's essential to identify food that is possibly bad. Bitter taste is generally triggered by poisons, but also by some other alkaloids such as nicotine or aspirin. And umami taste is triggered by proteins, things like cheese, beef, or pork. Let's talk now about the anatomy of an ear and then about the physiology of hearing. Ear has 3 distinct parts: outer ear, middle ear, and inner ear. The outer ear consists of pinna, that's the structure that we usually call an ear, and auditory canal or external auditory canal or external acoustic meatus. The function of pinna and external auditory canal is to capture the sound and direct them towards the middle ear. Middle ear, right here, and external auditory canal are separated by the membrane called tympanic membrane. Tympanic membrane vibrates in response to the sound, and these vibrations are transmitted to 3 auditory ossicles: malleus, which is attached to the tympanic membrane, incus, which is attached to malleus, and stapes, located between the incus. Right here's the stapes. Stapes is located between the incus and so-called oval window. And then stapes transmits the vibrations to the fluid that is contained inside of the inner ear. There is a tube called eustachian or auditory or pharyngotympanic tube that connects middle ear to the outside world. The function of eustachian tube is to make sure that the pressure in the middle ear and in our environment are the same, and nothing obstructs or impedes the vibrations of the tympanic membrane. When stapes vibrates, its vibrations are transmitted to the oval window. Here you can see stapes that is attached to the oval window. So vibrations from stapes are transmitted to the fluid inside of the inner ear. The cochlea, the part of the inner ear that is responsible for sensing for hearing sensations. Right here it's the cochlea. Cochlea in the inner ear has three important compartments: vestibular canal, tympanic canal, and organ of Corti located in so-called cochlear duct. Organ of Corti contains a whole bunch of hair cells. These hair cells have stereocilia on top of them. Stereocilia of the hair cells can bend in response to the vibrations of fluid in the inner ear. Essentially, the hair cells of the organ of Corti, right here, you can see right here, hair cells of the organ of Corti have their stereocilia embedded into tictorial membrane. When fluid in the inner ear vibrates, so-called basilar membrane, shown here in beige, vibrates as well, and that leads to the bending of stereocilia. When stereocilia bend, that eventually creates a nerve impulse, which is then transmitted to the brain as a sound. Now, interestingly enough, that in basilar membrane there are multiple fibers, multiple parts, and each part of the basilar membrane is sensitive to a different pitch. This is how we can determine what pitch of the sound there is. The louder the sound, the more intensive the vibrations are going to be, and that will be also transmitted to the brain. As you know, this is how we're able to distinguish sounds of different intensity. Now, the two types of deafness that can occur in the ear are conduction and nerve deafness. So, conduction deafness is when the transmission of the sound through the ear structures is impaired. For instance, tympanic membrane is pierced, or auditory ossicles are damaged or fused together. Conduction deafness can be ameliorated by increasing the sound amplitude via hearing aid. Nerve deafness occurs when sound cannot be detected by the cochlear nerves, or impulse is not generated by the hair cells. It can arise from the direct damage to the hair cells or direct damage to the cochlear nerve. And nerve deafness can be ameliorated by introduction of cochlear implants that directly convert the sound, the vibrations, into electrical impulses that are later relayed into the nerve. Now let's take a look what happens with the balance. So equilibrium can be static or dynamic. So static equilibrium is dealing with sensing horizontal and vertical linear acceleration. The organ that is responsible for static equilibrium is vestibule, which contains two separate chambers, saccule and utricle. So when one of them is responsible for vertical acceleration, one of them is responsible for horizontal acceleration. Utricle senses horizontal acceleration, while saccule senses vertical acceleration. Inside of both utricle and saccule, there is a receptor called maculae. You can see macula right here. In macula, you also have hair cells, and stereocilia of the hair cells is embedded into otolith membrane. When, for instance, your head tilts forward, otolith membrane tilts forward, bending the hair cells and generating nerve impulses that are delivered to the brain via vestibular division of vestibulocochlear nerve. Dynamic equilibrium is a rotational equilibrium. It senses rotational movements, and it is sensed by semicircular canals. Inside of the semicircular canals, you have an sensory organ called crista ampullaris. Inside of crista ampullaris, you have ampullary cupula, a gel-like structure which embeds in itself the hair cells. When your head is rotating, the crista ampullaris here bends in the position in the direction that is opposite to the rotation. This bending bends the stereocilia of the hair cells, and that generates the electrical signal which is transmitted to the brain via the vestibulocochlear nerves. Now let's talk about the eye. Eye can be divided, so the eye consists of, first of all, 3 layers: fibrous, vascular, and neural. Vascular layer consists of 2 specific parts. It's a cornea, and sclera. Sclera, also known as white of an eye. The function of cornea is the refraction of light to bring the picture into focus on the retina, while the function of sclera is to provide the site of attachment for extrinsic eye muscles, muscles that move the eye, to protect and maintain the structure of an eye. The vascular layer of an eye consists of 3 subparts: choroid, ciliary body, and iris. So the function of choroid is to supply the blood to the retinal layer of an eye. Ciliary body, shown here and here, controls the shape of the lens and also produces the aqueous humor that fills up the anterior chamber of an eye. The function of iris, I'm going to remove all the ink, start over. The function of the iris is to regulate the size of the pupil by dilating and constricting it. The third layer of the eye is a neural or retinal layer. The main part of neural or nervous layer is retina. The function of retina is to receive the light as a stimulus and transform the light into the nerve signals. The photoreceptor cells in the retinal layer are embedded into pigmented layer, which is essential for recycling the photopigment and prevent the light scattering in the eye. Spatially, eye can be divided into anterior cavity, which is confined between the sclera and the lens, and vitreous chamber, which occupies the rest of an eye. Anterior cavity is filled with aqueous humor, which is produced by ciliary body and drained into so-called scleral venous sinus, so-called canal of Schlemm. When drainage of the aqueous humor in the anterior cavity is impeded, then the fluid starts to accumulate into anterior cavity, which increases intraocular pressure, which leads to a condition known as glaucoma. Vitreous chamber is filled with the vitreous humor, which is essential for maintaining the shape of an eye. Interestingly enough, vitreous humor is formed when during the embryonic development and does not change throughout the life of an individual. Now, let's talk a little bit about the process of accommodation, process of how we focus the image in the eye. So, the image that we are looking at is focused on the retina. Now you can see that the shape of the lens changes from when we are looking at the distant object to the situation when we are looking at the close object. Essentially, when we're looking at the distant object, lenses are more flat, and when we are looking at the closer object, lenses become bulging. This change in the lens shape essentially is an accommodation. With age, accommodation becomes more different because multiple layers of cells are added in the lens. Lenses become thicker and stiffer, which makes harder for them to change their shape. Now let's talk about what is nearsightedness and what is farsightedness. In nearsightedness, the eye is anatomically too long and the image is focused in front of the retina. So when light rays hit the retina, they are all dispersed and fuzzy. It can be corrected by concave lens. In farsightedness, the eye is too short and image is focused behind the retina, which can be corrected by convex lens. In astigmatism, the problem usually lies in the cornea, which is shaped imperfectly, and that leads to basically combination, as you can see, of farsightedness and nearsightedness in the same eye. You essentially have multiple focal planes for the image that are not located in the retina. It can be corrected either by special lens or special glasses, or by the laser surgery. Now let's talk about retina. Vitreous humor that fills the vitreous chamber holds the retina in place. Retina is entirely composed of neurons. For instance, rods and cones are photoreceptor cells that respond to light. Cones respond to bright light and are responsible for high-resolution, sharp color vision in the bright light. Cones are concentrated near the center of the retina in the place called macula lutea. In the center of macula lutea is the area called fovea centralis, which essentially contains only cones. Rods can function when light is dim, when light is not bright. They, unfortunately, due to certain arrangement, although they can detect low light, they can provide only vague images. So they essentially are responsible for night vision and for peripheral vision. They are spread mostly through the retinal periphery. Now, I want to go back to cones and talk about that. So, first of all, the place that all your images are essentially focused on is fovea centralis, because it contains only cones, it has highest visual acuity, and provides the brightest and the sharpest image. Second, how do we see different colors? We have 3 different cone colors, red, blue, and green, and by combining those cone colors, we can form properly colored vision perception. Now, signals that are generated by rods and cones, they then relate to the brain via bipolar cells, and bipolar cells relate to ganglion cells, which essentially deliver the nerve signals, action potentials, from the photoreceptors to the brain. Now, when a photon of light hits the rod on the in the retina, chemical rhodopsin initiates the neural response. Now, rhodopsin is a special visual pigment. It can respond to low levels of white light. We mentioned that rods are responding to low levels of light. Light splits rhodopsin into two components, retinal and opsin. And eventually that split leads to generation of action potential. In cones, visual pigments are basically the same, retinal and opsin, but there are slightly different types of opsins in cones, which provide them with higher visual acuity and can detect red, green, and blue light. The signal from photoreceptor cells, as we mentioned, is related to bipolar cells, and bipolar cells synapse with ganglion cells. Ganglion cells, essentially the axons form optic nerve, which then delivers signal to the brain. In this image you can see the communication between cones and rods to bipolar cells to ganglion cells. So impulses that go through ganglionic cells and they enter the optic nerve, and you can see that optic nerve forms a blind spot here, the back of the retina. If you wonder why you actually do not see a blind spot in your eyes, because your brain compensates for that blind spot by constantly moving the eye around. Now, optic nerve enters the thalamus, and from the thalamus are delivered into the occipital lobe where the visual cortices are. So from impulses from each side, they reach into the same hemisphere and to the opposite hemisphere. They cross at the optic chiasma. So basically what you see with the right eye, these images are partially projected to the left side of the visual cortex and vice versa. The image that reaches your visual cortex is essentially flipped upside down in left and right, but the brain very early in life learns how to flip it back, and this is why you don't see the world around you in the upside down left and right flipped manner.